My name is Annina Schmid, and I'm a specialist musculoskeletal physiotherapist and an associate professor at the Nuffield Department for Clinical Neurosciences at Oxford University. My group uses a translational approach to advance our understanding of the pathomechanisms of entrapment neuropathies and neuropathic pain, with the ultimate goal to improve management for these patients. In this manuscript, which is published in Brain, we have explored the molecular and cellular determinants of nerve regeneration and their relationship to neuropathic pain in humans. Whereas neural regeneration has been extensively studied in animal models of peripheral nerve injury, it remains challenging to study the determinants of nerve regeneration in humans due to the limited access to tissues and a lack of treatments to promote nerve regeneration. Here, we use carpal tunnel syndrome as a model system to study sensory nerve regeneration in humans after chronic denervation. Carpal tunnel syndrome is the most common entrapment neuropathy and one of the few neuropathic pain conditions that can be treated surgically. It therefore represents a unique model system to prospectively study the cellular and molecular correlates of nerve regeneration from injury to recovery in humans. We have performed deep phenotyping in 60 patients with carpal tunnel syndrome before and 6 months after carpal tunnel surgery, which induces nerve regeneration. 20 healthy participants provided normative data. 6 months after surgery, a significant recovery of median nerve neurophysiological measures and somatosensory function was apparent. Serial skin biopsies taken in the affected nerve territory of the hand revealed a partial recovery of intraepidermal nerve fiber density 6 months after surgery. Intriguingly, the extent of intraepidermal nerve fiber density correlated with symptom improvement. So we asked ourselves the question whether there may be a molecular signature associated with this nerve regeneration into target tissue. To answer this question, we performed transcriptional profiling of serial skin biopsies and identified 31 genes that were significantly differentially expressed after compared to before carpal tunnel surgery. The most strongly dysregulated gene was ADCYAP1, which encodes pituitary adenylate cyclase activating peptide, in short PACAP. PACAP is a pleiotropic protein that acts as a neurotrophic factor, neuromodulator, and neurotransmitter. What caught our attention is that PACAP was not only upregulated almost fourfold after surgery, but its expression was associated with recovery of intraepidermal nerve fibers, and we found it expressed in skin sensory afferents. In order to confirm a regenerative function of PACAP in human sensory neurons, we moved to an in vitro model system. Specifically, we cultured mature human-induced pluripotent stem cell-derived sensory neurons, which we then dissociated and replated, mimicking an axonal injury, to study their ability to regenerate neurites. Excitingly, the application of PACAP to the human-induced pluripotent stem cell-derived sensory neurons increased their neurite outgrowth in a dose-dependent manner, confirming a pro-regenerative role of PACAP in human sensory neurons. A similar regenerative effect on human sensory neurons was induced by the selective PACAP receptor PAC1 agonist maxadilin. As you can see, our data from a unique human model system and confirmed in human-induced pluripotent stem cells in vitro identified ADCYAP1 encoding PACAP to have an important regenerative role in human sensory neurons. Manipulation of PACAP or its receptor PAC1 may have therapeutic potential in human sensory nerve regeneration.